Good afternoon. This session deals with market interest in oncolytic viruses. I'm Bob Martuzza from Mass General Hospital and Harvard Medical School. And with me as co-moderator is Martine Lamfers from Erasmus University Medical Center. And we have three individuals from industry, Anlong Lee from Merck, Jeffrey Infante from Janssen, and Loic Vincent from Takeda. So let me start with the following. Each of you is pursuing different oncolytic virus strategies. Please introduce yourself and your company and say why you think your choice of platform of oncolytic virus and strategy are optimal. So I'll ask each of you. So Jeffrey, why don't you begin?
Yeah, hi, Jeff Infante. I'm a medical oncologist and I head early development at Janssen. So we've been interested in oncolytic virus for years now, and the platform we've chosen, and I'm not sure it's the optimal one or not, I think the clinic will have to bear that out, is the HSV platform. There's been data with this, or precedent with it, with other with this platform, I heard the previous session, TVEC was mentioned, so we know that it is a platform that can work in patients. It also is a large virus and allows capability of putting multiple kind of cargo into it, as we're all, I think, trying to find ways to optimize these next generation of oncolytic viruses.
Very good. Anlong?
Hi, good afternoon. This is Anlong Lee. I'm a clinical director from Merck Early Oncology Group. Like Jeffrey said, we have this. We do not control which platform we choose. We actually acquired this oncolytic virus from a small company, biotech from Australia. You probably already know that this previously called Calibrating. Actually, it's also called CAV21. It's coxsackie virus A21 group. Actually, it's a common cold virus. We acquired from Australia since 2018, and we started develop clinical development from 2019. So currently, a couple of studies ongoing. We actually use it by intratumor injection or intravenous treatment combined with our PD-1 antibody. So I hope it will be interesting.
Very good. Thank you. Loic?
Hi, everyone. So my name is Loic Vincent. I'm the head of oncology research at Takeda Oncology. So at Takeda Oncology, what we are doing, we are advancing a portfolio of best-in-class and first-in-class immuno-oncology assets that are currently in preclinical and clinical stage. And core to our R&D strategy is our partnering approach, where we work with key academic institutions as well as biotechs. And in the field of oncolytic virus, we're working with Townstone Bio, that has been funded by John Bell, who is one of the pioneers in the oncolytic virus field. We have a first oncolytic virus that is in the clinic called TAC605. It's an optimized Copenhagen strain of vaccinia virus that is engineered to express different payload, FLT3 ligand, membrane-bound IL-12, and a full-length anti-CTLA-4 antibody. It's currently in phase 1, and we have additional programs in research where we're trying to use this novel optimized vaccinia platform to engineer and express different payload that can ignite the immune response. So as Jeffrey, I can't tell you it's the optimal platform. We still need to see the efficacy and safety and build translational package in patients. It's our preferred approach. It's differentiating from what others are doing, and we believe it provides patients with novel therapeutic avenues.
That's a great start to what each of you are doing. Martine, let me turn it over to you.
Yes, thank you. So, well, despite the promise of oncolytic viral therapy for cancer treatment, we've seen a relatively slow advancement of the field. Several hurdles have been proposed. For example, it's known that these oncolytic virus trials can be logistically and technically quite demanding with patient isolation requirements or advanced delivery techniques, but also the clinical grade manufacturing of the product, regulatory aspects, and more. They can all pose challenges. So the question is, what hurdles or challenges have there been in the development and implementation of your strategy? And do you think these hurdles have kept other companies out of the field? Maybe you could start, Jeffrey?
Sure. Thank you. I mean, it's a great and loaded question, honestly, because everything you said is truly a hurdle as we try to bring these therapies forward for patients. I think the number one hurdle though is to get these to work, right? Like those other things are real, and they're hard to do. But if these oncolytic viruses start producing systemic effects that really change outcomes for patients, then the field will adapt, right? And I think things will adapt. I think the reason people aren't in it at full force at this point, besides a few companies, is that it's been challenging to get these viruses to work. We see at the site of injection, they often do produce a lot of the effects that we're looking for, but for these to be real therapies, they're going to have to have an effect systemically at all areas of the cancer. And that, to me, is still the biggest challenge and what everyone here on the phone is trying to go after.
Yeah, so demonstration of efficacy actually as the major challenge or hurdle. And Long, can you also comment from your side?
Oh, yeah. For us, I know different companies have different challenge. For us, the major challenge is this virus currently is mostly given by intratumor injection. Not every cell you have the experience how to inject those drugs to the tumor. So especially if the tumor is in an organ like liver or gastric, it's kind of challenge for the site to do that. The small challenge actually is, the deliver and store the drug, because this RNA virus is need to store at -70 or -80 freezer, and deliver in a lower temperature. Yes, But I think we are trying to give it by intravenous, which is more convenient for the site can do that. We're currently still ongoing. So I hope we can go good efficacy in the future. If we can get good efficacy, we can systemically treat patients. So by intravenous, it should be much easier.
So the focus is on delivery issues and storage issues.
Yeah. Mm-hmm.
All right, thank you very much. Loic, can you also give your thoughts?
Sure. I mean, there's been a lot of things already said, so I won't repeat what has been told. What I have to say is that whatever oncolytic virus strain that you use, if you go with the intravenous route of administration, first of all, you need to make your oncolytic virus stable in human blood. They are immunogenic by nature. So what we are doing with the novel vaccine strain we're using with Townstone, it has been engineered to decrease the antiviral response against the oncolytic virus. Then when you're in the bloodstream, you need to evade the Kupffer cells. And then when you reach the tumor, you need to reach the tumor at a sufficient concentration so that it can elicit an oncolysis. And so far, the mechanism of action of oncolysis with the oncolytic virus has not been realized with the current strain. So everyone is using the engineering capabilities to make the oncolytic virus as a cargo for different payload that can orchestrate a broader immune response that can trigger this anti-tumor response in the tumor, as well as to different sites of metastasis. So I think why some companies have not yet entered the field is, as Jeffrey said, the lack of efficacy. You need to do randomized clinical trials, few hundreds of patients. It's going to take some time, and it's expensive, right? And if you go intratumorally, you need to work with interventional radiologists. It increases the cost of the procedure. You can decrease the market reach as well. So there are some challenges there. It's not like cell therapies where you can have conditional approval with single-arm efficacy with overall response rate. Here you will need to have event-driven endpoints, progression-free survival, overall survival. So it takes more time, more resources. It's a nice, I believe it's a nice approach to complement a diverse immuno-oncology pipeline that companies are advancing.
And do you think that these complexities of these trials are hurdles that are actually keeping companies out of the field, that are not making this step?
I mean, when you look at the investment of entering the field of oncolytic virus versus other, I would say, low-hanging fruit like cell therapy, right? You may think that you can have a cell therapy that can work in a specific indication. With oncolytic virus, most of them today are used intratumorally. So it's a challenge in terms of delivery. Once a company or group is going to come with an oncolytic virus platform that is not immunogenic, that is able to be delivered intravenously, that is able to have some oncolysis on the top of additional mechanism of action that you bring with the payload, there will be a boom in the field. But so far, it has not been achieved.
All right, thank you.
So this time, let's go in the opposite order. So Loic, I'll start with you, but then go to the others. So this field began with the concept of oncolysis, hence the name oncolytic viruses. But many groups are exploring various synergies, immune checkpoint inhibitors, pharmacologic agents, chemotherapy, miRNA modulation, and various others. So let me ask you, what areas excite you as looking like the best areas to go into in terms of something not only novel, but efficacious?
Yeah, I mean, that's a good question. Many things excite me in this field. I think you can see oncolytic virus as a great combination partner to really unlock the immune response. Of course, everyone is combining with anti-PD-1, but when you look at the engineering capabilities of oncolytic virus, most of the companies are doing that, having an oncolytic virus backbone that is able to express different payload. We are doing that with TAC605. We have different approaches that are in discovery stage as well. And when you look at diverse pipeline, the different mechanism of action in immuno-oncology, you can activate the immune cycle at different steps. And then you can combine with some other therapies that you have in your pipeline, including cell therapies. One can use oncolytic virus to change the tumor microenvironment and then unlock this tumor microenvironment to make the cell therapy work in solid tumor space, where we know the bar is relatively high. So it's endless in terms of engineering, which is also complicated because you need to make the right call, the right mechanism of action that you believe are the primary mechanism of action that you want to trigger in a tumor.
Very good. Anlong, your thoughts?
Yeah, I agree with him. I don't have any comments.
Yeah.
Yeah, I mean, I agree with everything Luke said. The one other area that excites me about these is, and it's kind of counterintuitive to our traditional model of development where you start in very advanced patients and try to have a response and then start moving in an earlier stage disease. But knowing the direct effects of these in the current state where we have to do intratumor injection with the majority of these, I'm interested in limited stage disease for these type of therapies, right? And so, we know that patients with early stage disease often relapse systemically. And if you could treat at the site of the tumor when it's relatively infant in its course, then maybe you get the immune response you need before the before the cancer becomes extremely complicated and hard to treat. So in addition to those, I think the excitement for me in this field is that you could move it pretty early into the disease setting for patients.
So two follow-up questions on the same topic. We would never expect one dose of chemotherapy to cure somebody or one dose of radiation, yet many of the studies, particularly those in brain tumors, are a single dose. So tell me what your thoughts are about multiple dosing. In other words, an initial dose, a booster dose, maybe more booster doses, et cetera. And then secondly, with chemotherapy, it's rare that a monotherapy is given. We're into 2 and 3 chemotherapies at a time. Do you ever see using two different viruses sequentially? So if you could answer, Jeff, we'll go with you.
I think you raise great points. I think that probably, if not the biggest one challenge so far is the need for intratumor injection. And anytime you're doing that and you want to do it more than once, it limits your patient population that you can test it in, because it has to be something that's really superficial on the skin or palpable. And the idea of injecting a liver lesion multiple times really doesn't feel good, and it's unlikely to be a successful track. So either these drugs have to work with the single injection, and we get them optimized correctly with the right payloads, and you create the effect you want, or we have to find other ways of doing it. You bring up the different platform approach that you could potentially put 2 together. I know this is a common strategy for vaccines, as far as like a prime boost is kind of the term we use internally for a vaccine, where you prime with one vaccine platform and then you boost the antigens that you want with a second vaccine platform. To my knowledge, that hasn't been tried in this oncolytic virus field today, but could be potentially feasible. I think you're still stuck with this intratumor injection thing right now, right? And Lok mentioned, if we can get systemic, then a lot of options open up. If you have 2 different platforms, you're probably going to be doing it sequentially, and it's still multiple injections, which gets tricky.
Well, I think with the intravenous, the thought might be that it'd be hard to give multiple injections because people develop an antibody response, whereas a second virus might not have that response until it was given times. Analog, your thoughts?
Yes, I agree. Yeah, I agree. So intravenous is not easy. Injection, intratumor injection is not easy. Lots of intratumor injection is focused on the cutaneous, subcutaneous. For those kind of cancer, we can boost the doses. Like us, we have few doses before the PD-1 antibody can used, so that actually maximum awake the immune system to kill the cancer cell. Eventually, I think we should still develop the intravenous treatment. So you cannot just use focus on IT intratumor injection.
Loic, for lack of time, I'm going to pass on you this time and go over to Martine.
Okay, thank you. In clinical trials with oncolytic viruses so far, some impressive durable responses have been observed, even in very highly malignant tumors. But in general, the response rates still remain quite low. Do you believe that patient stratification approaches may be required to increase these response rates? For example, stratifying for seronegative or based on the activation status of interferon response pathways or specific cancer pathways. So what are your thoughts on this? And is your company putting effort in identifying markers of response? Maybe we can start with Loic.
Sure. I think to my knowledge, there's no robust strategy to prospectively stratify patients and to understand which patient population is likely to respond to an oncolytic virus today. I think what we are doing comes back to basic science, understanding the oncolytic virus you're using. When you get into the clinic, when you generate data, those oncolytic viruses are engineered to express different payloads. So you need to try to assess the response that you see in patients, and then you can have some insight into what type of patient population is likely to respond or not. And then reverse translate with some fast follower approaches where you leverage the clinical learning in your preclinical stage oncolytic virus. I think what you said, stratifying for seropositive versus seronegative, is clearly something that is, I would say, easy to do with the oncolytic virus field, but the field is not there yet. We need to invest more energy on trying to understand what are the patient populations that are likely to respond to some particular oncolytic virus strain. I'm pretty sure the strategy for vaccinia can be different for any other oncolytic virus backbone.
Okay, thank you. Hello?
Yeah, I agree with him. Actually, for the early oncology, especially the phase 1 trial, we normally use all comers, but actually in the phase 2 or phase 3, it's different strategy. Stratification maybe is the best way to target the specific population, either use the biomarker or something else. If you see some response from the early phase, you probably need associated response with your biomarker findings. After that, you can stratify a patient population. Is not every patient got responded to those oncolytic virus for sure. So if you can choose the right biomarker, so you're gonna got targeted a specific population, the patient will get best benefit for this. I agree with that. Stratification is very important, especially in the late stage clinical development.
But it sounds like up to now, it's mostly retrospective studies that are being done. And that's also what your companies are working towards, to look at retrospectively in these trials to see who were responders and who were not.
Yes. Mm-hmm. Yeah.
I mean, you take that into account already when you design the trials, is that you have enough samples, tissue samples, whatever blood samples that you need to investigate and to reach any conclusions on this site.
Mm-hmm. Yeah, correct.
Okay,
Thanks.
Thank you. Jeffrey, do you want to add?
Yeah, I really don't have a lot to add, but I think if you're not thinking about a patient selection strategy, then you're probably not developing drugs correctly. But like the previous speakers have said, right now we just don't have the data to be able to do it from the get-go, right? And so you want to design your trials so you can answer that question as you're building it, and that's critical.
Thank you very much.
So various academics and drug companies are making more aggressive viruses. And what I mean by that is viruses expressing various cytokines that could have a cytokine storm, viruses that multiply better, et cetera. Is there a need to make these viruses with either a turn-off mechanism or some kind of modulation mechanism? I'll start with Jeffrey, because herpes already has that.
Yeah, I mean, there's a lot of interest in this field, and still multiple ways that it's going to be optimized and change over time. This is definitely one of them. We currently don't have a turn-off strategy, but I could see for other platforms that may be important. Say it again.
Valcyclovir.
Yeah, you could exactly, but we don't have to use it but maybe especially if you ever get to the stage where you can give these things systemically, then I really see where that could be an important thing moving forward.
Analog, what are your thoughts?
Yeah, I think it's correct. Actually, for our company, we actually not use genetically modified oncolytic virus. We use the common cold virus. Actually, it's a natural virus. For us, it's not a problem. I know the genetically modified oncolytic virus maybe need carefully to monitor the AEs in a very early stage. So like you said, the immune system. Need to be a cytokine storm. You need very closely to monitor the AEs and have the guideline how to treat this AEs.
And Loic, you use a large DNA virus that it lends itself to putting various inserts and expression genes in it. What are your thoughts on this?
So like others, we're not using turn-off mechanism of action. No safety switch. I think so far the oncolytic virus are safe. They have been tested in more 1,000 patients and not aware of major adverse events. We're using an optimized strain. We are the first with Townstone to test it in the clinic, and so far so good. And I think if we were to face some cytokine storm or any other immune-related adverse event, I think the field has evolved a lot and learned a lot from the cell therapies and the experience that physicians have in this field that could be applied to oncolytic virus. If we were to see this type of response, I would say that I would be, first of all, happy for the mechanism of action, because it means that this is power enough to induce a major immune response. Of course, we'll have to be clearly monitored with the physicians and maybe ICU experts.
So it sounds like everybody's taking the approach of if it ain't broke, don't fix it. Wait until we have a problem and then worry about it. Is that it?
Yeah.
It's still an early field where we have to learn.
Okay. Well, we're at the point of having to stop, unfortunately, and move on to the next session. But I want to very much thank Martine for being the co-moderator, and the three of you, Jeffrey, Ann Long, and Loic, for joining in. Thank you very much.
Thank you.
Wish you all the best. Thank you.